Okay, great. So, hi everybody. Welcome to our second event. I am super happy that you managed to be here. This session is also going to be recorded and we can share it afterwards. We are happy to have you here. We are the people from Neurarcane, an association that tries to spark the interest of students in neuroscience. So without any further ado, I will give the floor to our speaker.
Thank you, everyone. Thank you for introducing me. Neurarcane. My name is Christos Botos. I come from Greece, but you can just call me Chris. Chris from Greece, if you remember right. And I will talk to you today about epigenetics and gene regulation, hoping to maybe bridge neuroscience and genetics. And I hope you get some nice ideas out of it, and it sparks some desire for knowledge in this particular field. So, just some things about me first. So, who am I? My name is, again, Christos Botos. I have graduated with a bachelor's in molecular biology from the University of Crete in Greece. I have also been a research assistant for almost 2 years now in IMBB-FORTH, the Institute for Molecular Biology and Biotechnology in FORTH in Greece. Now, since I'm in the Netherlands, I'm working remotely, but I'm also a computational biologist, so I don't need to be in a lab to conduct my research. And I'm currently in the Leiden University Medical Center here in the Netherlands for 2 months now. I have been a research intern there, conducting research before I start my master's. So my IMBB work specializes in transcriptional dynamics. I simulate them in silico, so in the PC. And here in LUMC, I'm doing spatial transcriptomics. All right, so enough about me. Let's go to the presentation. So what to expect? Because this is a long presentation, I wanted to make it more interactive. So at some parts, especially in the beginning, I'm going to ask you some questions. I hope you're brave enough to give me your best guess, that you don't have to know everything. But an interactive presentation is more fun for you, for me, because this is a discussion between future scientists, right? And about the summary, what we are going to talk to you today about, we are first going to learn some things about the fundamentals of gene expression, how our genes are regulated and then turned into proteins that are expressed as phenotypes in our bodies. And then I will show you 5 of the key gene regulation mechanisms, mostly having to do with epigenetics. And then I thought of picking a particular gene you might be interested in. I don't know if you have heard of the enzyme called Tyrosine Hydroxylase. It's actually the rate-limiting enzyme for dopamine, the neurotransmitter. So I will talk more about that. I thought of picking a particular gene because neuroepigenetics is a vast subject. So focusing on a particular gene can showcase all these mechanisms in a condensed example. But I will also, at the end, also give the stage to some specific interesting neurogenetic findings and papers about other aspects. Again, if I didn't mention it, neuroepigenetics is the combination of epigenetics and neuroscience. So this is what we are going to talk about today. All right, let's start. So the fundamentals of gene expression. We're going to start with something that many of you might have seen, but if not, this is the central dogma of molecular biology. So in humans, the genetic material, so what holds the information about what we are going to become and look like, is in 46 linear DNA double-stranded molecules, so DNA, right? From that from those big DNA molecules, we go to RNA, which is like, let's say, the small, more erratic and energetic cousin of DNA. It makes small copies of it, and it moves around the cell, passing the information to particular places. So then it can be translated to proteins, which are the machines of the cell. They do particular jobs. So we go from DNA to RNA to proteins. You may ask, why is RNA needed? That's actually a really big question. But one explanation I can give you is that DNA is a particular part of the cell, and it's very big. It's not easy to move it around. It's easier for DNA to make small copies of particular parts of itself and set them around. But it's a more complicated question than that. All right. There are also some, so we have replication of DNA, then transcription, and then translation. There are also these mechanisms of reverse transcription and RNA replications, which are not found that much in humans. It's mostly microbial aspects. I will mention, though, for your interest, if you have heard of HIV, it works with RNA replication. So the main drug for HIV targets RNA replication. It stops that, and that's how it's battled against in the patients. All right. So after that, I want to ask the first introductory question. Don't be afraid to make guesses. How long do you think in humans the DNA is? So if we took all 46 molecules together and we attach them one after the other, how long would that be? Any takers?
3 meters?
All right.
The distance of the Earth to the Moon.
Both valid answers. Any guesses? Meters or kilobases? You can guess.
12 kilometers.
12 kilometers. Wow. You know you have trillions of those in your, right? But that's actually not a bad way to think about it. Any other guesses, or should I reveal the answer? So, it is 6 times 10 to the 9th kilobases, which is equal to around 2.2 meters. 2.2 meters of DNA in all of you. And not only that, but how many do you think you have? In your whole body, so each nucleus usually contains one DNA, right? One DNA, I mean, one copy of the genome, all right? How many nuclei do we have in us? 6 to 7 trillion nuclei. So 6 to 7 cells containing nuclei. How many are in the brain? 86 billion. And to think about size, think of a tip of a needle. 10,000 nuclei can fit on the tip of that needle. And each one of them has 2 meters of DNA in you right now. How is that even possible? Well, it's very long, as we said, but it's also very, very thin, just some nanometers. So you have a very long molecule that is also very thin, and you are trying to condense it and fit it inside the nucleus. This is how that looks like. So this is the DNA double helix, as we will see it, but it isn't alone. I don't know if you have heard of a concept called chromatin. Chromatin is the DNA, the proteins around it, and the RNA all together in the nucleus. So the DNA gets wrapped around these octamers. These octamers are called nucleosomes, and they're made from proteins called histones. And the DNA around, if I remember correctly, 146 bases of it, wraps around one nucleosome. And then the nucleosomes themselves make coils and farther condense. We call this supercoiling. And then those threads that get made by the nucleus also farther condense until you have the chromosome. So from the 46 DNA molecules that we have in our nuclei, they all make one chromosome. And it's super, super condensed. So as you can see, the DNA is not only its sequence, but it also has a particular 3D structure. But let's not move on from its size so quickly. It's 2 meters. Do you need all of that to make proteins? Actually, no. The protein coding genes, so the part of the DNA that is translated into proteins, is only 2%. Only 2%. Do you know what they called the rest, like 2 decades ago? Junk DNA. And they didn't know what it was for, so they just called it junk. But nowadays, we're starting to see a different picture. First of all, we have introns, which, from mechanisms I will explain later, also play a pivotal role. But also, the other parts of DNA can do many regulatory things. They can tell to the chromatin how it's going to condense, and how they can determine the 3D structure of the DNA. They can be promoters, they can be enhancers, silencers, other genes. I will call them genes, but anyway, other parts of DNA that are not protein coding genes, but they help regulate them. So a big part of science in the gene regulation part of biology is how these other regions that are not translated into and expressed into proteins, how do they determine what we are? So a new term was made called epigenetics. I'm going to ask you to be brave again. So has anyone heard of epigenetics before? Could you maybe try to give an explanation of what it is in one sentence? 3 words, whatever comes to mind. Any takers? Yes?
So we've learned about epigenetics a bit, and it's mostly about the interaction between environment and our DNA, and how those interaction changes the expression of your DNA. But that's pretty much what we learned about it.
I see. That's a very good answer, though. But so environment does change the epigenetic part and your epigenetic profile, and it comes to a thing called reversibility. So the environment can change epigenetics because epigenetics are reversible. You can go back to that. Unlike the sequence, because when you try to change the sequence itself, you have mutations. All right? But epigenetics is not only the environment and its reversibility. Any other takers? All right. It's also, have you heard of heredity? Like DNA can also pass on. The genetic changes in the DNA can pass on to your children. So it's not only the sequence that passes on to the next generation, but also epigenetic changes in in your gametes can pass on to the next generation. So epigenetics is the hereditary but reversible changes that happened to the DNA or the genomic material of that organism, which in humans is the DNA. Yes?
Does it change the gene itself or only affect its expression or suppression?
So when you say the gene itself, it doesn't change by definition the sequence. It can change its biochemical aspects, as I will show later. So maybe particular bases in that gene could change biochemically. You will see when we get to DNA methylation afterwards. Yeah.
So it's not the same thing as having mutations?
No, because first of all, mutations can be seen in the sequence. Genetics is way harder to actually see and determine. And also, mutations, when you have a mutation in one cell here, when that mutation happens, it will always pass on to after it divides, right? But with epigenetic changes, maybe there is an epigenetic change here, which is with white, but that could pass on to only one of them. Because during replication, there are specific mechanisms that may try to stop changes in the epigenome, in at least one of them. Again, I will show more when we get to DNA methylation about that part. But I hope it has piqued your interest. All right, so we move on. So these are the basics of gene expression. We go from DNA to RNA to proteins, and also the 3D structure plays a role. I will show you the first and most known epigenetic mechanism. It's also like the traditional epigenetic mechanism. Do you remember histones, which make up the nuclei? The nucleosomes, and those histones have DNA wrapped around them. Well, as it happens, those histones have a tail. So this is a tail extending out of every one of those histones. So you have 8 per nucleosome. And on those tails, we have seen that there are many modifications that happen, like phosphorylations, methylations, acetylations, ubiquitinations. And they happen at particular points, and they can have different effects. So one of them is that they can act as landing pads. So think of proteins like a helicopter, and those are they all have a specific like landing pad. So when they see a specific modification, they go there and attach themselves. So instead of having a protein moving around, you now have it attached to a specific tail. So if there is this is a nucleosome, and this is a tail. So if a protein, instead of going around and trying to find this specific sequence of DNA to do something here, if it finds this, then this can bring it here straight away. So you have protein effects happening faster. This, many of you may think of as a catalytic mechanism, like you don't actually change what happens, but you make it happen faster, right? So modifications can act as landing pads for other proteins. Those proteins can come here and they can do stuff. They can be transcription factors. They can, in other words, start transcription. They can stop transcription. They can move the DNA around and bring specific sequences closer. So if you have in DNA here and DNA here, and it loops, and you have these are 2 nucleosomes with the same modification here, a protein could come that recognizes both of them and brings DNA together. All right? So anything you can imagine, you will probably find it. But can those modifications, instead of using proteins, can they do changes themselves just from the modification? Well, let's see if you get a bit braver. These questions will be sparser as we go on. But I want to ask one more. Do you know if DNA is charged? Have you ever heard of that? In water, of course. Could you guess maybe what the charge of DNA is? Well, what's the full name of DNA? So DNA is an acid, deoxyribonucleic acid, right? So acid means, if you remember from high school, that it interacts with water and it makes a negative thing. So DNA is negative. And yes, of course, it charges the water as well. So here, the DNA is negative in water, and of course, us as humans and most living species have water in them, and the DNA exists in a water environment, so it becomes negative. It exists as something that is negatively charged, so it needs something positive to stabilize it. Well, that's where histones come in. Histones are mostly positively charged because they have many bases in their tails. All right. So histones are positively charged, and the DNA around it is negatively charged. That's how they are so strongly bonded together. Well, what will happen if I went to the histone and changed its charge. So a lysine, which is an amino acid, is usually positively charged, okay? If I go here and I acetylate it, anyway, if I go here and I acetylate it, it will become neutral. And when it becomes neutral, then DNA is more relaxed around the nucleosome. So what changes here? Well, as you can see, if I was a protein and I was trying to access DNA that is wrapped around the histone here, I couldn't get in. I'm too big. I have been losing some weight, but I'm still too big. But here, this is the time of my life. It's so relaxed around the nucleosome. So as a protein, I can go there and do my job at that specific sequence. Okay? So histone acetylation leads to proteins doing stuff. Usually, that means expression of genes. So when a gene is expressed, we usually see acetylation around it. So the chromatin is relaxed. This is what we call heterochromatin and achromatin. So here, in heterochromatin, the chromatin is very condensed. Most proteins are too big to go in there and do stuff, all right? So then we have F-chromatin, which is, let's say, the active side of DNA. And here things happen, and here we see acetylations on the histones. All right? And let me actually show you a very, very cool video of this. This is, so there is a thing called molecular dynamics, and this is a lab that used a supercomputer to actually simulate what happens. Before I started, here is a nucleosome. There are eight histones here, and you can see the tail is extending, right? Okay. So let's see what happens as DNA gets wrapped around it firstly, and then acetylations may happen. So here is the tail. You can see it's extending out like a hook, trying to find proteins. Here is the DNA. It gets wrapped around the nucleosome. You can see the tails in a specific color here. And then one of the tails can be acetylated. So the green light here is an acetylation, okay? So it's changed there, its charge changed, so the DNA will start now being more relaxed around the nucleosome. See the difference? Here, there are no acetylations, and here there are acetylations. Can you see how it's more compacted here and more relaxed here? So easier for a protein to come in and do some work. All right. So let me move this back and go back to my boring presentation. But this video was really cool in my mind. So what's different? Like, we all have those cells in us, right? They came from the same mother cell. But how is one a dopaminergic neuron and one is a neural stem cell? How is one a cell in our bones? How is one a cell in our kidneys? What's different if they all have the same sequence, the same exact sequence? Well, the 3D structure of DNA is what's different. So as you can see here, the same regions can be moved around and have different neighboring regions in different neurons and different cells in general. This is a neural stem cell, and it goes to a dopaminergic neuron. The reverse can happen, but it's a big discussion, and it's really difficult, usually. So we go from a specific, usually the higher in the hierarchy cells, like stem cells, have less epigenetic modifications. And as we go down to specific cells, the epigenetic modifications get more specific, all right? But yes, this is usually not always the case. So what changes? It's the 3D structure changes. Why do we care about the 3D structure? It can have any composition it wants. Well, it matters what DNA regions are close to other DNA regions in space, not in sequence. For example, there are specific regulatory DNA sequences that are called enhancers or silencers. I will call them enhancers for now. Silencer is the opposite of an enhancer. So enhancers boost transcription. They help in transcription. Silencers, as you can understand, do the opposite. They block transcription. So these two regions are really, really far apart, okay, genetically. But the DNA has looked around and it has brought them together in the 3D space. So this enhancer is very close to this promoter of that gene. Well, that enhancer can be recognized by specific proteins, as you see here, the green, blue, and the lighter blue. It can also produce RNA, but let's not go deeper into that, it's a bit rabbit hole. But it can be recognized by specific proteins, and those proteins bring in transcription factors, so proteins that help boost transcription. So it's a cascade where many proteins come together, bound by the enhancer, and they help transcription actually happen. So this is why we care. If that enhancer is not there, then transcription may not happen. That's actually, in a lot of diseases, what happens. So, all right, 3D space, histone modifications, enhancers, we got that, but there must be more, right? Well, the second traditional epigenetic mechanism is called, as I mentioned before, DNA methylations. These are specific methyl groups that come to, so methyl is this, and they come and bind to the DNA sequence, right? And they stay there. They stay there for a long time. There are many enzymes actually that come and put more there or remove. But if you don't have these enzymes, these can stay here for months. So it's a very stable change in the DNA. So that's why I said you don't actually change the sequence, but you can change biochemical properties around it. And yes, this can also be passed on to the next generation. If you have if your mother had methylation in a specific promoter, it's very common that you will also have that. All right. So DNA methylations, though, how do they work? Well, that's a big question. It mostly depends on where it happens. Mostly when we talk about DNA methylations, we focus on the promoter. And in the promoter, it usually stops transcription. So the methyl groups go there. Then they recruit specific proteins. So these are the methyl group. And they recruit a specific protein here, and this blocks transcription. For example, one of them brings together two nucleosomes, really, really close. So not other transcription factors can come in. Again, there are many, many mechanisms how this happens. If you can think of something, there's probably a case for that. So in the promoter, they usually stop transcription. In the gene body, usually they boost it, but we have found a lot of cases that, again, they stop it. So it is really context dependent. Why are they important? Well, in embryonic development, they're very important. Different cells that you want to maybe, you want these particular cells to divide more in a specific region, then the genes that have to do with mitosis will not be methylated, and then when you want that to stop, their promoters will be DNA methylated. Have you heard of X chromosome inactivation? So in women have 2 X chromosomes, right? Men have 1. Well, you have a whole second X chromosome. Where is all that's a big change, right? How are both men and women still human? It's one whole chromosome. Well, in women, one of the chromosomes gets inactivated. So, and it's not the same in every one of your cells. So different cells in you, if you are ladies, one of the 2 X chromosomes is inactivated. And how that happens? Well, we have found that the promoters of all the genes in that chromosome have all been DNA methylated. So, of course, there's modifications and other stuff that help, but methylation really helps in deactivating the X chromosome. And also, chromosomes, like you have them in your mind, only happen when the cell is close to dividing. So it helps bring them in a stable state, because when the cells start mitosis, you don't want them to actually still be transcribing and making RNAs and proteins. You want them to stop for a bit, do the deviation, and then start again. So DNA methylation is also important. Now, two more really important regulated mechanisms. Well, there are many different kinds of RNAs, many different ones. I decided to pick one of them to show to you today, which is microRNAs. You may know of messenger RNA, tRNA, and rRNA, which are the three main ones. But there are also other types, like non-coding RNAs, circular RNAs. And a very, very important one is, especially for neuroscience research today, it's microRNAs. And actually, it's a very, very studied right now. It's a very good way to see what may happen in one's body before it happens. I know many people who work on strokes for that. They measure miRNA in patients, and they can predict if they have a stroke or not. So it's a very active area of research. That's why I picked it. And the mechanism is very easy, don't worry. So you have the messenger RNA, right? It comes from genes, protein-coded genes. Then that messenger RNA leaves the nucleus. And if all goes well, it will go to a protein. But there is a microRNA, which is 20 to 30 bases long, not that long, but it recognizes a specific sequence in that messenger RNA. And if it finds it, it bonds with it because it's complementary, and then it stops it from actually being translated. And then slowly it gets broken down. So maybe you have a gene being transcribed and everything goes well, but before it reaches the ribosome, after it leaves the nucleus, it can also be stopped at that point. So yes, so this is microRNAs. They're really, really important. We will see some examples later in neuroscience. So last but not least, maybe you have heard of splicing. So there are introns and exons in protein coding genes. And alternative splicing, I wanted to mention it, is when those introns are not cut correctly, or those exons may be excluded. So not what you would expect to happen from most humans actually happens. So here, you can see that we have 3 exons, the 2 yellow and 1 blue, and sometimes the blue exon can be in there, can remain, sometimes it may not. And this happens, as far as I know, to almost all proteins. Almost all of them have alternative splicing. And you can find many different protein isoforms, and then concentration matters in what actually happens in the cell. All right. And why am I mentioning this in the epigenetic mechanisms? Well, they are controlled by epigenetics. So maybe you have a modification in the blue nucleosome, you can the one here. Maybe you have a specific modification that boosts this isoform, the left one. Maybe if that modification changes, then you only see the right one. All right. So these are the 5 gene regulatory mechanisms. All right, we have the 2 traditional epigenetic ones, DNA methylation and histone modifications, and then we have enhancers or silencers, and then alternative splicing and microRNAs. Well, we have these 5, right? But of course there are more, but these are 5 key ones I picked for today. Well, we're neuroscientists. I mean, you're neuroscientists. Good for you, and I admire you for that. But you may think, okay, those mechanisms are interesting, but how do they matter to me and my research? Well, I wanted to show some examples, but the field is so vast that there are so many examples of different genes. So as to not to confuse you, I picked a particular one gene, and I picked, I think, one that is very important in neuroscience research today. And this is the gene that is expressed into the protein called tyrosine hydroxylase. All right? This is the rate limiting. I will wipe away the hi, everyone. I already said hi. So, TH is a rate limiting enzyme for dopamine production, right? So it limits this step here. And the more you have, the more dopamine production you will have for a specific time frame, okay? So if you have the gene for this enzyme be more expressed, that usually leads to more dopamine down the road. If it's less expressed, less dopamine. Well, now for the last time, I would like to use your help, but this you can answer anonymously. You probably know dopamine, right, from your studies. Can you think of any neurological issues or phenomena that have a lot to do with dopamine? Have you heard of a specific issue that causes less dopamine or more dopamine, or a specific issue that is caused by less dopamine or more dopamine, and not only issues? I have picked 3 of them, but I would love to see your examples. Your QR code. All right. So let's check here. Some interesting guesses. See a lot of diseases too. That's a good start. But also think of, let me help, lifestyle maybe, and substances as well. Maybe you get something in your body. Sorry. Yeah. So one is a psychiatric disorder, out of those I picked. Another is an issue caused by lifestyle choices in our everyday life, and another maybe from something we ingest or put in ourselves. All right, so and let's see what you picked. It's here. All right, some very good guesses, and I apologize if your guess won't be spoken about today. It's a very good guess, but I picked 3 interesting examples. So I see a lot of schizophrenia and bipolar. There will be one of the examples I picked is about them. Many neuroscientists will agree that we see more dopamine in those cases. So in schizophrenia, in specific regions of the brain, we see an increase of dopamine. Then depression is a very good pick. I didn't pick it today because depression is also a very vast field. There are very different kinds of depression, but of course, dopamine probably does play a role there. Parkinson's, actually, I researched a lot on it. Actually, Parkinson's, for those who don't know, Parkinson's kills dopaminergic neurons, right? So we see a lot of less dopamine, but what happens to the actual gene for TH is actually not very well understood at the moment. So this is what we call in research a knowledge gap. Maybe one of you in the future can look into it. I found articles that say that TH is upregulated in surviving dopaminergic neurons. I mean, that would make sense, right? You have less dopamine, so the body tries to make more. But some others found it to be downregulated, maybe due to changes that Lewy bodies in Parkinson's make. Drug abuse is a very good pick. I also picked that. I will show you a bit more about cocaine use disorder. And so Parkinson's, schizophrenia, and ADHD. Yes, ADHD, as far as I know though, I saw some examples, but it's it's not that well studied as a disease yet. So I couldn't find particular examples for it, but I know that dopamine does play a role, and also again, this is a knowledge gap. So think about that for the future. So, very good guesses. Thank you so much. I hope I didn't miss anything. Let's go to the actual things I picked. And first, I would like to, I would like to talk to you about cocaine use disorder. And many of the things I will say here, I would guess that they're translated to a lot of drug disorders, drug use disorders, but I think cocaine, all right? So, let's say we have someone who takes the substance known as cocaine. Well, what does that do? Well, cocaine is known to stop dopamine reuptake from the synaptic cleft. So what actually happens is that it blocks the channels that usually bring dopamine back into the presynaptic neuron. And the dopamine stays in the cleft and has more time to trigger the postsynaptic neuron. But as you can imagine, this change does not only change that one particular fact, but it brings changes to the whole neural system because you dysregulated it. And as we talked about, everything is connected in the 3D space and everywhere. So how does it do it? Well, I picked a very interesting study that, first of all, a lot of studies show that a particular transcription factor called EGR1 is upregulated. Its gene then is upregulated in cocaine when you use cocaine. So we have more of this transcription factor, and this transcription factor goes to the TH gene, the tyrosine hydroxylase gene, and boosts its expression. Okay. Well, that transcription factor, as you can see up there, recognizes a particular enhancer. So that's how it works. What if something happened to that enhancer? Well, I found a very interesting study that showed that when you use cocaine not once, but chronically, as you will see, many of the epigenetic changes happen when you use something or do something a lot of times, giving it time to happen to a lot of your cells as well. Well, when you are a chronic user, they found that the enhancer up there becomes DNA methylated. So after it gets DNA methylated, the enhancer up here, again, those other proteins come in that recognize those methylations. And they block the EGR1 from going in there and actually doing its work and recognizing the enhancer and starting transcription. So you have DNA methylation happening here. Why? Well, it's not clear, but in many of these cases, the body tries to bring balance. So my hypothesis would be, and what they hypothesized in the paper, is that the body is trying to bring back homeostasis because it sees a lot of unnecessary expression of the TH gene. So it tries to stop that by methylating its enhancer. But what happens when you stop taking cocaine? Well, as I said before, those DNA methylations are very stable. They can stay there for months. Well, when you stop taking cocaine, the body has DNA methylated that enhancer, but it doesn't have cocaine boosting dopamine anymore. So you have less dopamine overall because you have less TH, less dopamine, as we said before. That's what we call withdrawal. Why does withdrawal happen? Well, epigenetic changes have already happened to counteract the dysregulation you brought on by yourself. And then when you stop and when you make another change and try to go back to yourself as you were before, well, your body is not that fast. So it takes time to slowly clear those DNA methylations. Can you also see another thing happening here as neuroscientists? This is a form of memory, isn't it? The DNA methylations stay there for months. So this is a way for the body to actually remember something without changing the genomic sequence at all. And actually, many of the models that they try to make for memory have a lot to do with epigenetic changes. Much of the memory we acquire throughout our lives may be saved in our epigenome, not only in the synapses themselves, which are also influenced by the epigenome. So a lot of interesting stuff to think about here. And I will also mention that there are enzymes that clear those methylations, and those are called TET. So I found another study where TET1 was downregulated during cocaine use disorder, and then upregulated afterwards during withdrawal. So the body is not oblivious. It does try to bring back and fix the DNA methylations it made. So it upregulates the enzyme that fixes them, but it's not super fast. It slowly happens, and it slowly happens across many different cells. All right, let's move on to what many of you guessed, which I also found a very interesting paper about, I would like to show you, with schizophrenia and bipolar disorder. Actually, maybe some of you could clear that for me, but I found that schizophrenia and bipolar disorder are correlated in a lot of studies. So many of the things that people found happening in schizophrenia also happened in bipolar. Maybe afterwards you can tell me your opinion on that and shed some light in my curiosity. So what happens with epigenetics here? Well, I don't know if you noticed before, but this enhancer, if you follow the DNA, so as to show that enhancers and all the regulatory sequences are not distinct as we would like to have them, this enhancer is inside the gene it enhances. If you can follow it, the enhancer for RG1 is inside the Th gene, in the exon 8, intron 8, and exon 9. So this whole region inside the Th gene is also an enhancer. So many of these DNA regions that we have distinct in our brains are not actually distinct, and they overlap, all right? Well, here, the enhancer is upstream, so it's before the gene. It's actually located within another gene. But here, again, we have the same logic. But instead of hypermethylated, in schizophrenia and bipolar, this paper found that the specific regions, another enhancer for Th, so this is a different enhancer, is hypomethylated for some reason. Probably what causes also the disorder, who knows? But they found that it's hypomethylated. So hypomethylation means that an activator, specific proteins that comes to that enhancer recognizes it easier and more often, okay? Because it doesn't find too many of these blocking in its way. So more of the transcription factor means more expression of Th. So they found that in schizophrenia and bipolar, an enhancer in the IGF2 gene is hypomethylated, and that leads to more dopamine in those regions. So that's an interesting area to study. Like, how does this happen? How does this affect? Is this found in every patient? All right. For the third and last example, and I think this is very interesting, I didn't see anyone guess it because it's not, let's say, that much of a disorder, right? It's a lifestyle change, I would say. Sleep deprivation, which many of us as students may have gone through. So sleep deprivation, what happens there? Actually, we have found that dopamine is increased in sleep deprivation. Maybe you can judge that by yourselves, right? Dopamine usually induces more stressful and fight or flight responses. It also makes us more impulsive and more in the moment. When you are sleep deprived, then you have rash decisions that you may make. Are you not more stressed sometimes? So they studied in rats that were sleep deprived without REM sleep for 72 hours. And they found that the microRNA, if you remember them, again, as you see here, they find the mRNA after transcription, they recognize it, bound to it, stop its translation, and they also lead to its destruction. So they found that a microRNA, this one, 335B, which is a direct mRNA of the mRNA produced by the TH gene, they found that in sleep deprivation, this is lowered, really lowered. So probably in sleep, what or one of the key players in the dopamine regulation is this microRNA. Well, the next step as scientists was like, okay, we see something happening, let's change that. Well, they injected this mRNA straight up, they injected it into the rats. And what did they find? Well, the rats stopped having maniac-like behaviors, like moving around with stress or being more impulsive. And the dopamine levels that they also measured afterwards were lowered and brought back to normal levels again. So this is a very interesting case of how microRNA can regulate what happens in us, right? So I wanted to focus on that particular gene, the TH gene, because many things happen around it, and also dopamine is a very important, a very well-studied neurotransmitter. But I couldn't not make some honorable mentions, right, in epigenetics, because there are so much stuff happening. Well, what I have here 4 papers. Neurarcane will give you all my sources and papers afterwards. You can check them out yourselves. And here we also see alternative splicing, first of all. So in learning and synaptic potentiation, alternative splicing plays a big role by changing the isoforms of the NMDA receptor, the N1. Anyway, that's its subunit, but the NMDA receptor. So that may change how this, if I'm not mistaken. Anyway, that may change how it works, right? How this receptor, maybe its different isoforms are less active, more active. They recognize a specific protein easier or harder. Also, they have found that histone acetylation methylations, as we talked about, are really important in synaptic plasticity and long-term memory consolidation. So let me give you an example for that. All right? Memory is very complicated, but one of the main ways that some people are hypothesizing memory working is the strengthening a specific like synapse. Right? So how could this happen? Maybe in the postsynaptic neuron, instead of, let's say, 2 channels for a particular neurotransmitter or calcium or anything, you go to 4. How? Well, the gene here, this gene, that was in heterochromatin, so it was very blocked by nucleosomes, this gene is responsible for making new channels, right? Or maybe bringing them to this area. And here, acetylation happens, freeing up this gene to make more of them, and then it closes it again. So you can see here how the changes. So this will be a stronger, let's say, synapse, right? So you can see how epigenetic changes here are linked to memory. And then we have many different kinds of RNAs as well, but what we learned today is microRNAs and how they regulate the neurogenesis, right? They stop, in this study, they found mRNAs that stop the mRNAs that are from genes that do deviations in stem cells before they become neurons, fully fledged neurons down the line. Finally, F-chromatin, so chromatin that is open, is observed in spinal cord regeneration. So I found a very interesting article where the spinal cord was broken, and they looked at what happens to the cells right before that cut. And they saw that it's very, very loose, the DNA. A lot of transcription happening, a lot of mitosis happening in cells that are higher in the hierarchy and can still do mitosis. And that's trying, the body is trying to repair what happened there. All right, so these were some honorable mentions. There are, of course, many others, and I would like to discuss them with you if you have any questions afterwards. So, for a conclusion, well, I don't know if you have heard of it, but all of this DNA and genomic and molecular biology revolution that we are now seeing happened only recently. Actually, in 2000, the cost of actually sequencing the whole human DNA was $100 million dollars. Do you know how much it is today? Like 3 years ago, less than 1,000. I think around today is around 500. For the whole 2 meter, as we said before, DNA, you can sequence that today for around $500. What 20, 25 years before you could do for millions only, and with many labs across the world collaborating. So this whole thing caused the genomic revolution. So we hear a lot about genes and mutations and how cancer happens from them, and how diseases are correlated to them, and how you can get from your mother or father specific genes. And we can now study all of that. We can get your whole family and sequence them and see what exactly has happened. But now that we learn more and more about genetics, we see a problem. We see a hole. The sequence, as you learned today, it doesn't give us the whole picture. Cannot say everything. So what's the next step? Well, a similar graph like this is probably going to be, in some years, the epigenetic graph. There are new ways, like there are many new ways, like Hi-C, which takes DNA and makes strong bonds here, and then you can study how close the DNA is. Those are covalent bonds. Or ataxic, if you have heard of it, it has a nice name. So ataxic goes to the different nucleosomes and it starts cutting, right? There are used specific attack nucleases that start cutting. And they cannot cut the DNA that's float around here because they cannot access it. They only cut here. So from how easy, then you measure like the different parts of DNA you get, and you see that many intact ones are probably around the nucleosome, and many that have been cut are free. So there are many, these are computational and molecular biology methods that you can now start studying the 3D structure of DNA as well, which is also very different between our cells, whereas the genomic sequence is the same across all of our cells. So that gives a high rise to another revolution right now. Everyone is putting it in their papers, which is called single cell. So now they're studying single individual cells. You get a specific neuron and that specific area, and you measure exactly what its epigenetics is and what happens to it. Because now epigenetics is different across many cells, so we have to go to single cell resolution. So, me as well, but also you, we are the young scientists of this next generation. And you as future neuroscientists, what does this mean for you? Well, in my mind, and maybe in yours, if I convinced you or showed you something interesting today, epigenetics and the 3D structure of the genomic sequence is the next step. Is the next step in research right now. And so I think it's very important, even if it's not your field, to keep it in mind and always see how it correlates to whatever you are studying or learning about or researching. And I ask all of you to be part of this next revolution in biology. And thank you for hosting me today. Thank you to Neurarcane, and thank you to all of you for listening.
Yes, so now it's time for the Q&A. So if you can just raise your hands and ask your questions, that would be great.
Today.
So I will start with the question. You mentioned about the memory of our body, and that sent me to the immune system, because I know that the T helper cells also learn. Is that also a kind of methylation, or is that related to the epigenetics?
Yes. Particularly, alternative splicing, a lot of it in there. So if you remember, alternative splicing is skipping over. So this is more to the immune system, but of course it's interesting. So alternative splicing again is maybe skipping an intron from removing it, or keeping an intron in, or skipping an exon. So whatever can happen there with the splicing. So maybe you have heard that T cells try to produce specific antisomes for a specific disease. All right. So if you have one gene here that stays stable, how do you produce different things to try them out? So how do you produce the antibodies here? So how do you produce them for this to be like white, and this to be black, and this to be squared? So this recognizes this recognizes this, and this recognizes this. So how do you make this change from one single genomic sequence? Well, you have here many different parts, and as the RNA polymerase passes over through them, alternative splicing can cut some of them out, producing variety. All right? So this is how it tries many different combinations to find the right one. But I will have to also note that apart from epigenetics, genetics also plays a role. What you actually see is that not only alternative splicing happens here, but also these move around and change place. And this is something I hid in my presentation a bit. I don't know if you saw it when I talked about transposons, but I didn't want to confuse you too much. But actually, yes, DNA can move around. So it can. Thank you. So DNA can actually move around. So it can a piece from here can move to here. Here can move to here. And not only just a bit of DNA, transposons with signs, lines, LDR retrotransposons and DNA transposons are 45% of our DNA. So 45 of them is transposons that can move around, but not all of them are active. Many of them are from viruses, many of them are selfish DNA sequences, so they don't give anything to the body, they just try to ride on with us. And around, I think, 10 to 5% are active, but a lot of DNA is moving around. So those two things happen here. The genomic sequence does change, but there is also epigenetic mechanisms like alternative splicing. Very nice question, by the way. Anything else? Someone was interested.
Maybe I will
Yeah.
ask a question as well. Excellent presentation, by the way. And we can clearly see that epigenetics can change our lives, potentially in the upcoming decades. I have a question about how easy is it to assess the amount of methylation or acetylation in specific tissues in vivo, meaning like in living beings, either animal models of certain diseases or humans.
Yes.
And the second part of the question is, how could it be improved in the future?
Yes, that's a very nice question. So this is a bit more on the methods, but how do we actually see those methylations and acetylations? Because they're really, really small. And methylation, again, is 4 molecules and not big ones at that. So how can you catch this? Well, actually, specific antibodies have been made that recognize histone tails that bear a specific modification. So you have your nucleosome here and the DNA wrapped around it. and it has one of its 8 tails here is, let's say, methylated, right? Well, you can use the nucleases I talked about earlier, so you can cut around. So you cut everything that isn't around the nucleosome, because small proteins come here and they cut, and what remains intact is only the DNA they can't actually access because they're too big for this area. Well, after that, you get many sequences that all have a nucleosome in them, but there can be different nucleosomes. So, for example, this can have acetylation, this can have ubiquitination. So, how do you find the one of those sequences that you want, the methylation? Then you can use specific antibodies that recognize this one, and then you can take the sequence of DNA and from the known genome map it. And then you say that in the whole 2 meters of DNA, I had this here, 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 and maybe here a bit more. But you may have noticed a problem in all of this, right? For humans especially. This is all well and good, but it requires me cutting my own DNA. Well, when we do that, we usually kill our model to test that, whether it is cells or animals. But to actually see them and keep them alive is a very difficult task, and it's where the research is actually heading right now. A lot of people are trying to use, in humans especially, adenoviruses, because they don't cause too many other effects. To actually put specific sequences in there. So maybe they put a sequence that has this antibody in there attached to a specific fluorescent protein, so it lights up. Maybe it could recognize this, but still there are, so it can go here and it can give you light in a specific area. But still there are many challenges here. You need very good microscopy because again, this is all very small. The DNA is very thin. You need to make sure that you don't affect the target too much. And there are also other approaches I may not know about or I may not have mentioned here, but the problem now is not just actually seeing the epigenetic changes, but seeing them without killing or affecting the model. Yeah, so is that...
Yeah, thank you so much.
Right. Any other questions that you have maybe to expand upon what I talked about here?
I have another question. So why did we evolve with epigenetics instead of just DNA? It's super complicated and DNA is super complicated. So why do we even have epigenetics?
Well, a very difficult question. But we can find, first of all, epigenetics in the roots of multicellular life. So when we started having many cells next to each other, bacterium or archaea or any other cell that's not eukaryotic do this as well. They bind together closely. So what's different between me and them? Well, they may bind and maybe they have some small changes between them, but they're mostly the same and they're mostly selfish. They want their own genes to be passed over. My neuron here is not selfish. It doesn't want its particular genes to pass over. That's called cancer. It cares about the genes I have in my cells that I'm going to give and make children from, right? Gametes. So my neuron has given up its ability to procreate for another cell that it hopes has the same sequence as that, because they are copies. So when we go to multicellular life, what happened is that some cells became less selfish. So instead of having the same one here, they all agree, okay, we agree that number 5 here, is the one who is going to pass on all of our genes and to the next generation. And 5 can focus only on that. It can keep mutations away, it can stay stable, and the rest of us can multiply and try to help 5. So instead of all of them trying to pass their genes, they gave up on control to help it. Well, for that to happen, you need differentiation. Because they all have the same genetic sequence, you have something to differentiate them by, and that's epigenetics. It's really interesting how it happens to the embryo, because it all comes from one cell. It usually has to do with the mother. The mother controls usually the epigenetics of the embryo. And then you have many genes trying to help one, many genomes in many different cells trying to help one, and you want different proteins activated in each one. I think you asked something else as well, or is this one?
No, this is pretty much my question.
All right.
But my next question would be...
I would like to say, yes, this is what I forgot. It's a very controversial topic if humans are better because we think better. But in the brain, a lot of the epigenetic mechanisms in humans are more complicated. So I'm just throwing it out there. It's a big discussion. But for example, if you remember the TET enzymes that come from alternative splicing, and they remove methylations here. They remove methylations like in the cocaine, right? But in humans we have 4, TET1, TET2, TET3, TET4. In primates, they only have TET1, TET2. In mice, they only have TET1. So a usual thing we observe is that epigenetics are more complicated in more complicated minds as well. But that's I'm walking a thin line here. So, yeah. Any other questions?
I have a question. So you said that the epigenetic makeup of the embryo is from the mother.
Yes.
So
Usually.
Usually. So can it come from the dad? Let's say you have the most healthy mother ever, and the dad is a smoker. He does a lot of stuff that can negatively impact his epigenetic marker. Can that affect the embryo as well?
Yes, but at what stage? So the dad's epigenetics can affect the embryo at the starting, like when the zygote is made, right? So you have a specific epigenetic makeup here in the sperm cell. This is a sperm cell error. I apologize. And you have a specific, in the egg of the mother as well, you have a specific epigenetic makeup as well. So these two combine, and they, in that way, what the father has done before, of course, smoking mostly changes the epigenetics of the lungs, but it can also affect omics. So father can affect this stage, but after this stage, when these two become one thing, then it's mostly about the mother and environmental factors as well. Yeah.
Can we know our epigenetic profile? So can we, when we look at our genetics, can we even look at our epigenetics?
Yes, in many ways. Not all of them having to kill the subject.
No.
Yes.
So can we know which diseases we might get, or the vulnerabilities or the strengths that we have?
Yes, as I mentioned before, to expand upon it because I found it actually very interesting. I have a colleague in Leiden who is studying, who is a neuroscientist in the neuroscience department of Leiden University Medical Center, and she's studying miRNAs, right? So miRNAs are interesting because that step of gene regulation, if you remember, happens after the RNA leaves the nucleus, so inside the cytoplasm. That means that mRNAs can escape everywhere. You can get a blood sample and find mRNAs that are found in neurons because they have escaped. So you can use that as a biomarker. So mRNAs are really hot right now as a biomarker. Many people are using them from blood samples or other samples around the body that do not kill the patient or affect, of course, too much. And they can, from that, measure miRNAs. And that particular colleague I mentioned, for example, has looked at correlations between strokes. So maybe an upregulation of a specific mRNA can alert doctors that a stroke isn't coming in the following days. So that's one way, and many more, probably. That's what comes to mind right now. If any of you have other ideas as well, I would love to hear them.
Is this how they're doing the testing for diseases in embryos as well, or that's something else?
In embryos, you can actually find some stuff about genetics. So the usual therapies they did, because epigenetics is more recent. Genetics is also recent, from what you saw from the graph, right? So right now, I think you're referring to the test that they do, where they actually get embryonic cells. They go in, scoop out specific embryonic cells, and they look at the genomic sequence. They can look at the epigenetics as well of those cells, yes, but those cells are of a particular variety, and epigenetic changes a lot with cell type, right? So if you wanted to study the brain, you will have to collect specific cells that differentiate down the line into the brain's cells. And that may be, I don't know, but that may be more difficult to collect from an embryo. So for genetics, yes, in embryos. For epigenetics, it probably depends on what you are trying to see, and some specific cells around the body could maybe be predicted easier than others. Yeah. Yes.
I have a question about DNA methylation too.
Yes.
You said that DNA methylation is very long-lasting and it's hereditary. So if I understand correctly, let's say my mom has a methylation at a specific area in her genes that can be transferred to me, right?
Yes.
How exactly does that happen?
So that methylation will have to take place in, first of all, not whatever cell your mother may have. For example, if a person's mother smokes, again, this could affect only the cells around the lungs, but it has a probability to also affect the gametes. So you want those particular cells pass on to the next generation. So you want to see the methylation there, right? As we said before, we only care about this one cell, and the others are supporting it. So how does this happen? Well, DNA methylation by itself is regulated by, first of all, by itself is very stable, but it's also regulated by these STEP enzymes, right? There are specific enzymes that try to keep it in place. So it's not only the methylation itself, it's also the expression of those enzymes. So it goes a step before and a step before, and many of them are feedback loops. But yes, an methylation will happen there, and it will also not happen in all of the cells. So maybe there is a chance, all right? And also, many of those genetic changes can also happen early in the embryonic development. For example, twins that are the exact same, right, from the same sac. They have the exact same genetic sequence, but they differ. They are very similar, but they differ in their genetics. And that may happen early on. Of course, down the line, they also get many environmental changes, and of course, with alcohol and stuff like that, they can change. But even early on, an embryo could be in this specific side of the sac and the other here. So an epigenetic change that happened from an environmental factor or electromagnetism from the mother or hormones from the mother could affect only this part of the embryo here, but not affect this other one. So, so it can happen to the mother as well. It can happen early in the embryo development as well. And it's one of the reasons twins are not the same, that are from the same cell. Do you want to?
Yeah.
All right, thanks.
So is then the embryo more sensitive to what's happening in the environment from a genetic point of view than the adults? Because for adults, for example, you need repeated alcohol use in order to...
Way more.
Yeah, but in embryos, they're way more sensitive?
Yes, because for two reasons. First of all, most of their cells are higher in the hierarchy. By the way, when I say higher in the hierarchy, I hope I didn't confuse anyone. I mean that the hierarchy of differentiation, right? So if down here is a dopaminergic neuron, and here is a stem cell, and here is like a neural stem cell. As you go from here to here, a lot more differentiation happens, and it's way more difficult to go back. So an embryo, concentration-wise, will have a lot more of those, and a lot more of those, and way less of those. And we, as developed humans, will have more of those. Right? Why is this important? Because the second reason is the number of cells. This does not go into one cell. This goes to, depending on the steps, 2, then 4, and many more. So a change in the embryo happens in one cell, and down the line, it leads to a change in 4. A change in us happens in one cell and stays in one cell. So it's number and level of differentiation, I would say. Yes?
Also, another question would be if a bad lifestyle, so I don't know, if you drink, for example, alcohol, is that going to influence it more and affect you more than if you're doing a good thing, like, I don't know, mindfulness, to be able to not be so stressed? So are the bad lifestyle factors affecting you more than the good lifestyle factors, or kind of the same?
Well, it's a very complicated question, right? And it probably differs from case to case. I can only make an educated guess. I would say that bad factors are probably more, they affect you more, because you already are in a good state. So you already have homeostasis. Everything is already working together. Making it work together better could not change too much, but breaking is like a machine, right? Breaking one part can collapse the whole machine, the whole car. Let's say you have a car and you have an engine, and let's say you get a new, really good part for that engine. But then one of the other part, how much does this improve the engine? A bit. Let's say now that instead of a good one, you go there and remove that part completely. Maybe it breaks down the whole engine. So I don't know. My educated guess will be, since we already have a good working system, improvements could cause some improvements in the phenotype as well, but doing bad stuff is a lot more disruptive and important.
Yeah, so then it means that we technically could also improve through epigenetics. So how far does it go? Like, can you change the color of your eyes, or your IQ, or your height, or what can you change with epigenetics?
Well, let me focus for simplicity's sake, because it's a very, very complicated stuff, of the three you mentioned, right? So color of your eyes is something that is not too epigenetically important, because it comes from a single gene. So there's one single gene that determines the color of your eyes. But for height, or IQ, or length of arm, or tallness, many of those things are what we call multi-gene dependent. So there are many unknown, probably, and many known genes that all affect your height, let's say, right? You could, for simplicity's sake, say that this gives you like 0.1 centimeter, this gives you 0.2, and maybe this 0.2 becomes from a mutation 0.1. So for color of your eyes and other things, it's one single gene. Usually the genetics is what matters. Here, it's many different genes, and many of them might be closed by epigenetics, many of them might be open. So here, epigenetics plays a much bigger role, I would say. So for changes, let's say you want to change the color of your eyes. This is caused by a mutation. But let's say you want to make them colorless, or maybe activate color in a patient that doesn't have melanin. Then you could go here and make this open again, or closed. So here it's a lot easier, because you are closing or opening a single gene. If you want to affect your height or your IQ, as you can imagine, it's a very complicated system. It's very difficult, but it's also very interesting. Because if we have, in the very future, an exact manuscript, we exactly know how each one gene, each one enhancer and silencer affect each other, then we will go to that level as well. For now, this is only possible with gene therapy. Yeah.
I have a question. So, how does epigenetics link to cancer? Like, a lot of like smoking packets say that smoking causes throat cancer or lung cancer, but how does that specifically work? Like, are there some genes that are silenced? Are there some protective genes that are, I don't know, not working anymore, or are there some genes that are turned on, that are some, like, I don't know, genes that are not necessarily helping us? Like, what's the mechanism behind it?
That's a very good question. Thank you very much. I didn't include cancer as you saw in my diseases sections. I know neurons can get cancers, but the cells around them that still divide can. But cancer and epigenetics right now is a very big question, right? Because there is so much evidence linking cancer to epigenetics, and it's one of the reasons why now cancer research is happening a lot in single cell. If you go to the National Cancer Institute, the Netherlands Cancer Institute in Amsterdam, you will see every lab doing single cell, because you want to know how one cell differs to the next in epigenetics. So cancer is caused can be caused by mutations. Let's say, for a simple example, you have a gene here that you have a gene here that is usually after the cell doesn't want to divide anymore, it has an enhancer here, right? And this activates this gene, and this gene causes mitosis. Well, when this cell may not want to divide anymore, it's stable, it may move this enhancer away. But in a cancer cell, you could get a mutation where, I don't know, many different things, but let's say that this a mutation in this enhancer, only this gene, manages to attract another protein that brings them together. So this is a model. You have a mutation in enhancer. It is recognized by another protein that brings them together, and then this enhancer enhances transcription there, causing mitosis. Okay, so there are many examples where both mutations, but also epigenetics afterwards can cause cancer. But why do they do single cell in cancer? Well, the bad thing about cancer is that it's very difficult to kill because it changes very much. So cancer cells usually have F chromatin, a lot of open chromatin, because they want to be able to transcribe a lot of stuff and be flexible. And if you try to here, let's say this is a cancer with 6 cells, and if you have an epigenetic profile here on those 4, which produces a particular protein, then you will go with chemotherapy and attack this particular protein with a drug and kill those 4. But cancer makes itself unstable, so here the epigenetics for that protein could be different, so it's not expressed here. So we see so much differentiation in cells in cancer because they try to protect themselves from both you as a doctor and your immune system. So epigenetics plays a big role in both how cancer is caused and how it survives afterwards. Cool thing we mentioned, elephants and big animals like whales don't die from cancer. How? They have so many cells, probably a cancerous one becomes. Well, one of the main hypotheses, this is a fun fact, is that they get so much cancer and they're so big that the previous cancer here, well, it's so unstable epigenetically and genetically that another different cancer is caused here and the second cancer kills the first. It's a very interesting fun fact to say that cancer is very unstable and has so much differentiation between the cells. All right.
Could we induce more cancer in humans to kill the previous cancer?
Yes, not we could, but it's actually a question that was asked before. But humans are very small compared to these animals, so you don't have too much space to work around in that. So the thing about these animals is that they are so big that then this big cancerous tumor cannot really kill the animal yet. It gets even bigger. And until that happens, a smaller one comes here statistically and stops it. But yeah, it's still an interesting question, I would say, but a bit sci-fi for now.
And why exactly smoking is causing cancer? So how does that interrelate with the epigenetics?
Well,
If you-
it correlates with genetics first, because cancer causes a lot of mutations, right? So again, a mutation can cause epigenetic upregulation of a gene you don't want. But cancer, but smoking, I did see some articles, I'm not completely sure, but I did see some articles that they can make epigenetic alterations as well. For example, as with cocaine, that I showed, when you take a substance, much of the time the body tries to bring back homeostasis. So in smoking as well, there may be epigenetic differences that stop specific genes that are activated by smoking. That's why you have also withdrawal in smoking. It's the same as cocaine, but yeah, it's probably a nuanced subject. All right.
So I also have another question.
Yes.
You can still go.
Yes.
So you mentioned during the slides with the sleep deprivation that there is some microRNA injected.
Yes.
Are we also able to do that in humans, or maybe some pills or something? And can we just get rid of the effects of sleep deprivation in that way?
Well, maybe, who knows? But again, sleep is very complicated. Many will say, and treat this as a disease, and say that you have a lot of dopamine in sleep. Let's bring that down. In sleep deprivation, sorry. Let's bring that down and treat it as a disease. But the body has a lot of reasons for much stuff. Maybe this dopamine be causing you impulsiveness and stress, but maybe it also helps in other aspects of sleep deprivation that we don't know about yet. So we need to be careful when we try to see everything around us as a disease and a disorder, because the body has many mechanisms why it does the stuff it does, and we only just started learning about them. But possibly.
We're not yet doing it in humans, changing the epigenetics chemically.
Bioethically, mostly. You can, of course, right now do that very easily, like methodologically. But the bioethics, because of our limited knowledge, stops us there, I would say.
So it's more of an ethical reason not to do it?
Yes, I mean, it's like you did it in rats. You can get the same microRNA injected in yourself. Nothing stops you, apart from bioethics. So, of course, you have, if you want to do this research, you have to get bioethical admission. But also, the rats are very similar to humans, but also it may not work in humans. Who knows? So,
But your educated guess would be that it's going to work in humans as well?
I mean, when you see something working in a model that is very close to humans, like mice, you would say that it may work on humans, and that would be a good hypothesis. It's not an unreasonable hypothesis. But still, you have some different. As I said before, for example, mice have only one, and we have 4. There are differences still. Yes?
I will come back to the previous topic about cancer. You said that till now they only worked on single cells. Are you aware if recently they developed any targeted therapies to confront cancer, and if it was any successful?
Gene therapies, you mean? What therapies?
Yeah.
Yes. Well, I'm not. I don't recall a particular paper that they have tried. I know they have tried in mice. You probably so for mice they have tried much stuff. They have tried putting in specific gene therapies in mice, and not only changing the genome, but also playing with epigenetic steps. For example, if you see like in cancer cells, this particular gene that causes mitosis might be up, then you would administer maybe miRNAs that target its particular mRNA, right? But so in mice they can do that, but this has so much, because again this gene exists in other cells as well that you want them to replicate. So when you stop replication, when you do chemotherapy, why does your heart fall out or your nails? Because those are cells that replicate very rapidly. So when you do a therapy that stops replication, mostly in cancer but all around, then you also see less replication here. But they have tried gene therapies, and it's very interesting in mice. In humans, again, due to bioethics, it's hard. And again, cancer is very unstable. So you may try a therapy here, but then it may be written over or overwhelmed, or the cancer cells can find their way out of it. Because with the gene therapy, you're targeting a particular subset of those cells, and there are still some that may overwhelm the other part because of their instability, and they're unstable and they do mitosis very quickly. Does that?
Yeah, thank you.
So we have time for one more question. One last question. If not, I will go. So how much can you actually change ethically with the lifestyle changes in your epigenetics? So,
Well, it's a new research area, but I would say much. Your diet, for example, can change a lot about your genetics. And especially, for example, as neuroscientists, maybe you are interested in the neuronal the gut system, right? So a lot of changes happen there from diet. And especially, for example, with the reward systems, I can maybe you eat, for example, a particular food. Has this ever happened to you where you like one food so much, you eat it every day for the next 10 days, and then you hate it? You hate it so much. Why? Well, an example would be that here in your gut, there is one neuron that was feeling this food and was sending you reward signals. But, and that happens again, like in the cocaine example, with one gene that produces a protein that maybe recognizes this food. But after you have too much of it, so your body always tries to bring balance back, maybe again it methylates, enhances or the promoter of this protein, making you unable to recognize this food this much, and then, or maybe the opposite can happen as well. It can send you bad signals. But yeah, again, your epigenetics and diet are very connected, and any substance, I would say, that you put in yourself, alcohol, smoking, of course, radioactive stuff, but any substance will make many changes. But not only substances. As I showed with sleep deprivation causes many genetic changes as well. So lifestyle, I would say, also matters. Our cycles, for example, our sleep cycle is very heavily modulated by epigenetics, genetics.
so then if it's that important, is the food industry regulated and keeping track of the epigenetics, or the pharma, big pharma, are they keeping track of that or not yet?
Well, not yet, I would say. Of course, there are efforts happening, but you have to look at epigenetics like something that happens with lagging before genetics. So, as you might have seen the past 10 years, they have started, now that genetic research is really cheap, they have started to look at genetics first. So things that cause mutations are regulated now. To go to the next step of epigenetics, I would say, just like before, give it another 10 years for them to also start looking there. So in my mind, I cannot predict the future, but I see epigenetics as following a similar model of what genetics has followed since 2000, what we have seen. Yes.
So as health recommendations,
Yes.
we've heard a lot in the media or Instagram or whatever about which lifestyle choices we should have. So what actually works? So you said sleep, for example, is really important, or
Yes.
the diet. What would you recommend?
Well, it's a very, I do not want to give advice for a particular lifestyle because many things work. I can say an educated guess from what we saw together. As you saw, the body with epigenetics lags behind the changes you made in your lifestyle. So you made the change this week. You are living life differently than what you did before. You recognize that, but your body slowly starts changing your regulation to appeal to that lifestyle. That's why when you make changes, for example, you go from one particular diet to another, you have many side effects. But then your body slowly, maybe due to epigenetic mechanisms, as we saw today with Leiden, maybe slowly starts getting ready for that lifestyle. So what our body likes is a stable routine, I would say. But that's not to say that you should keep a stable routine your whole life. But what I can make an educated guess is that, yeah, your body lags behind. So when you make changes, expect some kickback for a bit. That's it.
Also have a question. Is there like a specific time frame until you can see the kickback, like homeostasis wise? Or is it just like
very dependent on the number of cells that you are trying to see the change in, particular change that you want to see. Some changes may be irreversible, although I know said that epigenetics, its big part is that it's reversible, but after 2-month differentiation, some changes become irreversible, so you should be more careful, close when you're younger, and then you have more space to change your lifestyle and do bad stuff down the line. So that's what I would say to you, but that's the question. All right.
Thank you very much.
Thank you all for coming to my talk. All right.
Also, if you have further questions, our speaker is still here. So
yes, if anyone wants my contact information for anything, if you are interested also in research in Amsterdam or Leiden, you may talk to me. Yes, thank you very much.
Thank you everyone for coming.